everyone, I am Dr. Sayani Bandyopadhyay, Assistant Professor of Biology. Today we will discuss about DNA as the genetic material. It is the identification of DNA as a genetic material in the molecular basis of the information transfer. So the genetics is the study of heredity and variation, and DNA is the hereditary material. The deoxyribonucleic acid is the hereditary material in all the living organisms, and it carries the genetic information. So genetic information which transfer from one generation to another generation. Structure of DNA, we already heard that DNA has a double helix structure. So double helix model proposed by scientists Watson and Crick. It is a sugar phosphate backbone also, and the nitrogen bases are adenine, guanine, thymine and cytosine. So this is the structure in the nucleus. Inside the nucleus there is it's for eukaryotes. In prokaryotes there is no nucleus. In the eukaryotes there is a nucleus. Inside the nucleus it is chromosome, and in the chromosome fiber the DNA exists. So DNA replicates in the nucleus, and DNA is converted into RNA also in the nucleus in the process called transcription. So there are some experimental proof where DNA as a genetic material. First, in 1928, scientist Griffith demonstrated a transformation experiment. Then their experiment was further confirmed in 1944 by the scientists Avery, McLeod and McCarthy, and then finally DNA as a genetic material was confirmed by scientists Hershey and Chase in 1952. First, the Griffith's experiment. It is an experiment to understand how bacteria causes pneumonia in mice. It is an experiment for proving the transformation. So scientist Griffith, at first we will see the diagram. He used different strains of Streptococcus pneumonia bacteria. One is the R strain, which is called rough strain. Another one is the smooth strain. So the S strain or the smooth strain causes disease. So it is virulent. It causes pneumonia. And he used this, injects this strain in the mouse and into the mouse. And R strain is the rough strain, which is the non-virulent or harmless. So injection, at first live A strain was injected into the mouse and the mouse died. Then live R strain injected into the mouse and the mouse lived. Then the heat kill A strain were injected into the mouse. Previously we said that the A strain causes a disease in the mouse, so the mouse died. But the heat kill A strain did not cause any disease, so the mouse lived. And the heat kill A strain plus live R strain, which causes the death of the mouse. So individually the heat kill A strain did not cause any disease in the mouse, and individually the R strain did not cause any disease in the mouse. But by combining these two strains, the disease of the mouse was observed and the mouse died. You can see that first injection of rough strain, mouse lives. Injection of smooth strain, mouse dies. Injection of heat kill smooth strain, mouse lives. And injection of R strain plus heat kill smooth strain, so the mouse dies. Why this has happened? Because he concluded that this was because the R strain had somehow been transformed into the heat kill A strain. So he explained that was due to the transforming principle from the A strain to the R strain, which made the R strain virulent. So previously the R strain was not virulent, so the mouse lived. But then when it is transformed, then it becomes virulent and the mouse died. But it was not confirmed that it was DNA which is transformed. Then the experiments of Avery, McLeod and McCarthy. It is to identify the transforming principle from Griffith's experiment. So it is a further proofs of the Griffith experiment. They used the heat kill A strain, which was previously used by Griffiths, and they added some enzymes which is protease. Protease actually degrades the protein, RNase which degrades the RNA, and DNase which degrades the DNA. You can see these are the different proteins. One is proteinase which degrades the protein, RNase which degrades the RNA, and DNase which degrades the DNA. So what he had done, he individually added these enzymes into the this S10 extract. So this is the heat killed S10 extract. If he added proteinase at first, then after that he observed that S cell appeared. Then he added RNase and also observed that the S cell appeared. But when they added DNase, they observed that no S cell appeared, because if there was the heat killed S10 which was transformed, if that was protein, so after adding the proteinase it will be degraded. If it was RNA, then after adding the RNase it can be degraded. But if it was DNA, then after adding the DNase enzyme which degrades the DNA, then also the stem cells will be degraded. But here you can see the cells are degraded, so no S cells appears. So it was proved that this transformation requires DNA, which is actually transformed. So that's because it was DNA, so it is transformed, and so it is degraded by the adding the enzyme DNase. Last experiment was Hershey and Chase experiment. It is to determine whether DNA or protein is the genetic material in the virus. So they used that bacteriophages, which infects the bacteria. It is a virus. So they used labeled DNA of phages with phosphorus 32p32 found in, which is labeled with DNA, and the labeled protein code with sulfur S35, and allow the label first to infect the E. coli bacteria, because bacteriophages infect the bacteria. And observation, it was observed that after in the P32 DNA coated label entered the bacteria, but the S35 protein label remain outside. So it is the DNA which is actually entered the bacteria. So they concluded that DNA is the genetic material which transferred from the virus to the bacteria. So this is the experiment. In the green color we can see that it is a P32 labeled DNA, and then after transformation, so this is the phage genetic material, and this is the bacteria. So you can see that after the transformation you can see the green color. So green color means it is the DNA which transferred, but the S35 label protein code, which is the red color, but after infection you can see there is no red color, which means that the protein code is not transformed, but the DNA is transferred. So DNA is proved as the genetic material. So the packaging of DNA in prokaryotes like E. coli, which do not have a well-defined nucleus, so it doesn't contain any membrane-bound organelles like other prokaryotes. Here the positively charged histone proteins and the negatively charged DNA are held together in a loop, which is called nucleoid. So nucleus is made with positively charged histone protein and negatively charged DNA molecules. And in eukaryotes, the DNA is present in nucleus. So in nucleus there is a structure called nucleosome, where the negatively charged DNA is wrapped around the positively charged protein histone octamer. You can see this is the octamer 1, 2, 3, 4, 5, 6, 7, 8, and this is the histone, and this is the DNA which is wrapped. So we can say that DNA is a blueprint of life. The discovery is revolutionized biology and medicine. Ongoing research continues to unlock its vast potential in genetics, health, in biotechnology, as well as in evolution. Thank you so much.